this video, we are going to talk about the process of mitosis. First of all, let us see what is mitosis. Mitosis is a process of cell division which occurs in the living beings. At the end of this process, there are two daughter cells which are formed. Each cell contains an exact number of chromosomes and the genetic material as that of the parent cell. Here I have an image of a cell just to show you what are the different components which are present in a cell. The process of mitosis which we are going to study today has 5 main stages, and they are prophase, metaphase, anaphase, telophase and cytokinesis. Before the cell undergoes the process of mitosis, there is a stage which takes place known as interphase. In interphase, we have 3 different stages, that is G1 phase, S phase and G2 phase. In G1 and G2 phase, the cell growth takes place, and in S phase, the DNA synthesis takes place. At the end of the interphase, the cell which is supposed to undergo mitosis has all these things present in it. First is centriole, then microtubules, then we have nucleus. Inside the nucleus, we have nucleolus and chromatin, that is replicated DNA. Now let's talk about the stages of mitosis in detail. The first stage which we are going to talk about is prophase. Earlier we had talked about interphase, in which we talked about the centriole and microtubule, and we had seen that earlier they were in the condensed form. In prophase, these centrioles and microtubules, they start pairing up and they formed a centrosome as well. You can see over here the nucleolus has disappeared, the nuclear membrane is there, and inside the nuclear membrane the chromosomes, they start condensing. Before we move to the next step, that is metaphase, there is a phase known as late prophase. So over here the nuclear envelope, it breaks down. The paired centrioles which were formed earlier, they get placed at the opposite poles. Since the nuclear envelope is not there, so the microtubules, they go and they get attached to the center of the paired chromosomes. The next step is metaphase. In metaphase, the microtubules, as you can see over here, they get attached to the chromosomes, and the chromosomes, they get lined up at the metaphase plate. The third step is anaphase. In anaphase, the microtubules, they pull the chromosomes towards the poles. Now, from where do these microtubules, they pull the chromosomes? They pull the chromosomes from the center of it, known as centromere, as I have shown above. The last stage is telophase. In the previous stage, that is anaphase, we had seen that the microtubules were pulling the chromosomes apart. So once the chromosomes are completely moved towards the poles, the reforming of the nuclear envelope takes place. The chromosome, it starts decondensing and the polar microtubules are moved apart. This image over here, it explains what happens during the telophase. The last stage is cytokinesis. In this, as you can see over here, a centrosome is formed, the nuclear envelope has developed completely and the chromosomes are decondensed. Contractile ring is the site where the cell, it completely divides itself into 2 daughter cells. This image, it gives you a summary of the stages of mitosis and the cell division. This image, it gives you a recap of the stages of mitosis in detail. Advantages of the process. It is important for the growth, repair and regeneration of the tissues in living beings. Now let's talk about the disadvantages of the process. First one is no diversity. Since the daughter cells which are produced have identical chromosomes, so there is no diversity in them. And since the weaknesses and the traits are the same, so there is also a chance of extinction. If you have liked my video, do like, share and subscribe to my channel. Thank you.